Hello everyone. Here I'm going to share our lab's experience with optical genome mapping in evaluation of genome-wide structural alterations that include both copy number changes as well as gene fusions in myelodysplastic syndromes, or MDS for short. I have no relevant conflicts of interest to disclose. So structural chromosomal variants are important for diagnosis and prognostication of MDS. And in some cases, detection of specific structural variants can alter therapy. Unlike somatic mutation profiling, high throughput detection of structural variants has not been possible because of the presence of multiple repeat sequences in human genome. So right now, most well-characterized chromosomal alterations in MDS as well as AML are identified using a combination of traditional cytogenetic techniques. So these include conventional karyotyping, which has limited resolution because it's based on chromosomal banding patterns that is manually reviewed using light microscopy. FISH studies is more sensitive, but it has limited utility because it's a targeted approach. Chromosomal microarrays, they have a higher resolution and precision, but this assay is essentially ineffective in detecting balanced events such as inversions and translocations. Optical genome mapping, on the other hand, can potentially overcome this problem and provide a single platform for accurate identification of genome-wide abnormalities, in fact, all different types of structural variants at a fairly high resolution. So we decided to explore the utility of optical genome mapping in myelodysplastic syndrome. So why myelodysplastic syndrome? Now, and how important are structural variants in clinical management of myelodysplastic syndromes, or MDS? Now, based on the WHO classification guidelines, the diagnosis of MDS is dependent on recognizing dysplasia in at least 10% of cells in any of the three lineages. That would be granulocytic, erythroid, or megakaryocytic lineages. The morphologic assessment can be quite challenging, especially if the dysplasia is subtle or if the specimen is of limited quality. Now, on such diagnostically challenging cases, identification of specific cytogenetic abnormalities or structural variants is helpful to make the diagnosis of MDS. In fact, the structural variants that have been listed on this slide are considered as presumptive evidence of MDS in any patient with unexplained cytopenias. In some cases, the specific type of structural variant can guide treatment decisions. For example, the use of lenalidomide in MDS cases with isolated del(5q). Now, structural variants are very important in MDS for risk stratification. Now, despite identification of several gene mutations and molecular aberrations, the outcome of MDS is really and primarily dictated by the presence of larger chromosomal aberrations that are picked up by one of these techniques. Now, the treatment decision in myelodysplastic syndrome, whether it be to initiate the treatment or to change the therapy, it starts with risk stratification using the R-IPSS or revised IPSS scoring system. This includes blast percentage, the degree of cytopenia, as well as the structural variants. These include not only the number of structural variants, but also the type of alterations and encompass both the copy number changes as well as translocations. At this time, the abnormalities for R-IPSS are detected, the R-IPSS uses only the abnormalities detected by conventional karyotyping. So in our lab, we chose to explore the utility of optical genome mapping with the following objectives. So the first is OGM able to detect known structural variants that we are able to pick up using traditional technologies? Second, are we able to uncover new aberrations that are clinically relevant in MDS? And thirdly, do these new aberrations alter the outcome in MDS in clinical trial setting? So we chose to explore the utility of OGM in two phases. So the first phase was our proof of principles pilot study, and the second phase is where we are testing several samples of MDS. We've tested nearly 50 samples so far, and it's an ongoing project. So first, for the proof of principle study, We tested 12 samples of MDS, 9 with cytogenetic abnormalities, and 4 were diploid karyotype, 4 samples of acute myeloid leukemia, and 2 samples of acute lymphoblastic leukemias. Compared to conventional karyotyping, we had a concordance of greater than 90%. Compared to the microarray testing, we had 100% concordance. In addition, the only reason for the lower concordance in karyotype was because of the inability of the OGM to pick up 2 small aberrations that were seen within a setting of composite karyotype, thereby not meeting the limit of detection criteria. Now, in addition to the abnormalities detected by these traditional technologies, OGM was able to identify additional aberrations that are of potential clinical significance. Now, for this proof of principle study, we used extremely stringent criteria to identify the clinical significance. One of these criteria included that the segmental alteration involved one or more of the myeloid genes that were implicated in any of the myeloid neoplasms. And of this cohort, all of the additional aberrations of potential clinical cases importance were confirmed using alternate technologies, confirming the value of OGM. Since then, we have tested nearly 50 samples, and we have analyzed several of those, and we have confirmed the ability of OGM to detect all types of structural variants. And I want to highlight some of the important information that we're able to obtain from OGM using some examples. So here, the OGM is able to identify copy number gains and losses in autosomes as well as sex chromosomes with complete concordance with conventional cytogenetics and microarrays. Here we can see there is a large segmental deletion of chromosome 20 that the OGM is able to pick up that matched with the conventional karyotype. Now here is another example of MDS where we are able to identify deletions in chromosome 5 shown as in red. We are able to identify precise genomic details of the deletion as well as in addition, a gain of chromosome 6 that is apparent. Now, this was not picked up by conventional karyotype. In addition, as we noticed, the conventional karyotype shows deletion of chromosome Y. So when we look into it, although the circus plot, the deletion of chromosome Y is not that apparent, we are able to appreciate that in a copy number plot where the CNV of chromosome Y is less than 1 compared to a random control sample from an unrelated male. Now here we are able to see that the OGM is able to identify translocations between 2 different chromosomes, in this case chromosome 2 and chromosome 20. And at the same time, we were able to map out using OGM a complex chromosomal event. Here, it's a derivative chromosome that involves segments from chromosomes 1, 12, and 5. Now, using karyotype, this level of complexity involving this derivative chromosome was not apparent. Now, OGM was also able to show the breakpoint heterogeneity in genomic aberrations that is not apparent by conventional techniques. Now this, as we can see here, there are 2 different sizes for the chromosome 20 deletions, and we can also appreciate the 2 different fusion products here that translate to the deletions with of different sizes. Now, this is important for us to pick up because the clonal heterogeneity is responsible often for the resistance and relapses in hematologic malignancies. Now, the OGM is also able to clearly detect and map out the inversions, and here it's able to map it out at the gene level. This was an example of MDS with inversion of chromosome 3 that involves the MECOM or the EVI rearrangement, which is considered as a poor prognosis. Now, this level of gene detail essentially nullifies the need for an additional FISH assay to confirm the involvement by MECOM. Now, similarly, this is another MDS case which had a translocation 911 that was detected by conventional karyotyping. By OGM, in addition, we were able to identify a large deletion involving chromosome 11, and that encompassed the MLL or KMT2A gene. And this we were able to confirm by FISH and mapback FISH as well. Now, more importantly, OGM is able to uncover submicroscopic or cryptic aberrations that are clinically relevant. And these are often cryptic by other standard of care technologies. So this is a case of MDS, where we were able to detect a TET2 deletion involving chromosome 4. This was cryptic by karyotype because it's generally very small, and the labs do not routinely perform FISH for this specific deletion. Interestingly, this case by next generation sequencing studies also had a mutation in SF3B1 and an additional frameshift mutation in TET2, thereby confirming the biallelic inactivation of TET2. The point of this is essentially that all of the aberrations, except of course the mutation data, were obtainable using one platform or technique, avoiding the need for multiple confirmations. Now, this is also important because we are aware that the biallelic TET2 alterations are pretty frequent in myeloid neoplasms, and the deletions of TET2 are essentially missed if we do not perform a high throughput assay for structure variant detection. Now, to summarize, optical genome mapping essentially provides a single platform for accurately identifying all different types of structure variants, thereby avoiding the need for multiple confirmatory assays. Now, this makes it faster and less expensive overall. And in summary, I believe that this technology has immense potential to serve as a high throughput platform for structure variant analysis in the clinical laboratories. And I wanted to end the talk by acknowledging everyone that's responsible for implementing OGM in our lab. That includes Dr. Guillermo Garcia Monero, as well as Hui Yang, who's been hard at work on the bench, as well as my chair, Dr. Medeiros, and Dr. Lutra, who's leading the Molecular Diagnostics Lab. Thank you.